Hello Antonella, come stai?
Tutto bene, grazie.
Grazie bene. Okay, so I'd like to welcome you to this second seminar I have with the students. These students are mainly second and third and fourth year medical science students. So and they have a brief introduction in immunology. When I say brief, basic and diagnostic immunology. So I'm expecting very good questions from these students. Unfortunately for them, I've been teaching most of them. So let's see if my teaching has brought any fruits. Yes, Antonella? I would like to welcome you, of course. And I guess we can start straight away. What do you think?
Yeah, perfect. Yes. Yeah. Okay, I'm going to share my screen.
So we've practiced this, so hopefully the screen will show up very clearly now.
Can you see the slides?
Not yet. Not yet. So I think you need to choose.
Now?
Yes.
Okay, cool.
The mic is yours.
Perfect. Thank you very much. And thank you all for inviting me to discuss with you what's happening in Italy. As you know, we are in a very serious situation. So I'm glad we will be able to discuss the science behind this. But first, I'm going to give you an overview of the events in the last 2 weeks in Italy. I have to have a disclaimer, sorry. The following slides are intended for informational purposes only, and they are not meant in any case to substitute for medical opinion and care provided by physician or licensed medical provider. So first, I'm going to tell you a bit about my background. So I am a scientist, and I have more than 10 years experience in research. I worked both in academia and in industry. I did my degree in medical biotechnology at the University of Milan. And I worked with projects about innate immunity first, and then during my PhD, I also worked on adaptive immunity, so working on the role of complement in CD4 T cell biology, and especially at the interface with mucosal surfaces. I then moved on to some industry experience. I wanted to leave academia somehow and wanted to really experience the life cycle of drug discovery and development. So you can see here an infographic. What does it involve to create a medicine for patients? So there are several steps, and I can tell you I worked mainly in target validation, so where we identify a molecule and we want to target that, and then we progress on to lead discovery, lead optimization, and then preclinical evaluation, mostly done in animal models. So I worked a lot with murine models of respiratory infection. So having said that, so I would say my background really includes both academic and industry experience. I could experience the pros and cons of each of them. And also lately I started collaborating with Oxbridge Biotech Roundtable. This is an organization that aims to bring science and industry closer, because as you know, it is needed that the ideas that are generated in academia really find an application. So for that, we need industry collaboration. Moreover, I felt as a personal feeling that I wanted to be closer to patients. I think most in academia and in industry, as a scientist, you feel that your job is a small part of a big whole. And I felt personally that I was not close enough to patients. And lately I've been questioning myself whether I still want to be a scientist, or maybe I want to create something, a service, a company that can bring effective solutions to make the life of the patients easier. And then COVID-19 came as a bomb in our lives. And I have to say, all these thoughts that I had became real. We need to be close to patients. In this very moment, all the communities are becoming one, because patients are endangered and we need to protect their lives. So I selected here some papers and also videos, which I think can build the grounds to understand the specific aspects of COVID-19, which is the disease we are trying to fight. In particular, I would recommend this video. I hope you can see my pointer, which is made by a small company called Ninja Nerd Science. And here they explain really carefully epidemiology, pathophysiology, and diagnostics. Because I'm not a doctor, so I wanted to understand really how these patients, how do these patients present at the clinic? What are the clinical features? What do we need to help them with, etc. And then I'm gonna talk a bit about Italy. Luckily I am here in this moment. My job is in the UK, but I moved back a few months ago. I decided to quit the job in the UK and I'm here. So I have to say there was a big confusion in the beginning. We were probably underestimating the dimension of this problem. Some people were claiming is only a flu, is very similar to a flu and it will go away in a week or so. It's not this way. This is not a flu. We immediately understood that this was a serious, much more serious situation. So in terms of procedures that have been put in place by our government, there was an escalation of the so-called DPCM, which means Decree of the President of the Parliament. So around beginning of March, our Prime Minister on television said we need to limit the movement of people, limit the movement and the freedom. So this was a first step after patient one was discovered in north of Italy. And then it escalated with another DPCM. They said Lombardy, which is a region in the north of Italy, is a red zone because the contagion was spreading very quickly there. But also he said, I declare all Italy red zone. And in this moment, there was a big chaos. Basically, irresponsible people, I show you on a map to let you understand the extent of this. So the disease started spreading in this area. When the Prime Minister said all Italy is red zone and the movement has to be limited, some very irresponsible people started moving south. So basically bringing the virus in the south of Italy. These people included students, which is somehow understandable. They wanted to go back to their families. But also some people thought this was a holiday. So they decided to go back to their second houses they have in the south of Italy, where there is sun and sea and they enjoy their holidays usually. So you can tell this is very irresponsible behavior. Also considering that the hospitals in the north of Italy are way more equipped than the hospitals in Sardinia. south of Italy, and also especially Sardinia. Sorry, something is popping up. This is where I'm based, so we are an island, somehow protected by the fact that we are surrounded by the sea, but many people still made it to Sardinia until our governor decided to close both flight and ship traffic. So, patient one, very strange situation. So it is a guy from Codogno, a town in the north of Italy. So he declared that he met a Chinese colleague. He is a manager in one of the biggest companies in north of Italy. So they met for dinner, and then this guy started developing symptoms. It was later discovered that the Chinese person that he met did not test positive for coronavirus. So, many people started being judgmental and racist against Chinese people. This is not the case, because the virus doesn't care about race, where you come from, it really doesn't care. So it is not clear how patient one got the viral infection, but nonetheless, he was hospitalized in very serious condition. And he was hospitalized for nearly 3 weeks, and it's news of a few days ago, he made it alive, he recovered, and he will be able to meet the daughter that is soon to be born in the next days. So from the discovery of patient one, literally the disease spread with extreme speed in the north of Italy. And what happened was that elderly people, immediately we understood that elderly people were really in danger because they were hospitalized on a daily basis, lots of them, and many people started dying. So the towns which were most hit were Codogno, Bergamo in the north of Italy as well, Brescia. And to make you understand the extent of this disease, which is not a flu, so many people died that at a certain point there were no coffins anymore. They couldn't bear all this load of dead people. And so the army had to come and transfer the dead bodies from one town to another to be incinerated. So very sad. As I said, I was very confused in the first days. I couldn't understand how this is possible in 2020. Was there something we could have done to prevent it? Anyways, I think it is not the time for doubting and asking too many things, but it's the time of acting and fast. What was clear through the news, we knew immediately Italy was not ready, really. We didn't have enough swabs for PCR testing. We immediately realized personal protective equipment was not enough to protect doctors, and also we were running short of respirators. These patients come in and their situation escalates really quickly, and they need to be put on respirator support, oxygen support. So you can also understand that if there are not enough swabs, what it happens, some patients are not identified promptly as infected with the virus, even though they are hospitalized, and they pass the infection to doctors and nurses. There was a time doctors even complained, we can't go to war without personal protective equipment. If we fall ill, how can we help others? So another major problem with our situation in Italy. Also, people complained about the fact that the government didn't clearly explain the personal protective equipment and who needs them, because some people in the general population started buying a lot of masks, even the FFP2, FFP3, which are the ones that really function to avoid the infections. And by doing so, they were basically taking away this protective equipment from doctors. So a good direction from the government in that sense would have been helpful. About the respirators, so immediately Italians started thinking, how can we improve the numbers of the respirators? Also, a lot of crowdfunding campaigns started. We need to give money for the hospitals to buy everything they need. And having said that, you know, you can't imagine buying things in emergency where also the transport of things is somehow compromised. So many people started thinking, we are not ready for something like this. Our National Health Service is not ready. And they started thinking, this is very important and we need for future events to be ready with this. So hopefully a lot of awareness started building up within people. So the things that did work, fortunately, so social distancing after a problematic start, because I told you these people moving from north to south, it started working. And I don't know if you saw many captions online, they really explain why this is effective. I'm gonna show you again a caption here. So, okay, so you can see here, this is how the virus spreads. But if we practice social distancing by working from home, don't go to that barbecue, etc., then you start reducing the spread of infection. So Italians, I think, are generally anarchic people. They don't really like to be told what to do. So it was really hard in the first days, and then fines have been put in place, and we are talking about 3,000 euros fine if they find you, for example, going from one town to another without an important reason. So, so another good news, they started testing some drugs. So for example, I cite here tocilizumab, which is an anti-IL-6 antibody. So they received the approval, because of course you need to receive approval to test things onto patients. And they started seeing that somehow blocking IL-6, which is secreted in enormous quantities in these patients, these drugs started dampening somehow the inflammatory response that can be seen in these patients. Also, some other clever ideas. Doctors empirically started seeing that the prone position in patients that had their lungs severely compromised was helping and facilitating oxygenation. So I can imagine doctors trying whatever they can to help the clinical condition of the patients. And moreover, in Emilia-Romagna, which is another region of north of Italy, because they were running short of respirators, then they found a way to help 2 patients from a single respirator just by adding an additional tube. And this was a very clever idea to basically double the amount of respirators available. Unfortunately, a lot of fake news started becoming viral themselves on the internet, but also interesting theories that make us hope that science is on it, and soon we will receive the answers that we need. I added here, for example, the work of Mihai Netea, which is an immunologist from the Netherlands. So he started claiming that one possible explanation of the fact that really few children get the disease is because they are fresh from their vaccination calendars. And somehow he claims that innate immunity might be trained to fight even unknown pathogens, because the vaccinations themselves do train the innate immune system. So a central role also for the innate immune system, not only the adaptive one. And finally, I wanted to cite a video that was seen really as an explosion in Italy. Basically in 2015, a science newspaper that has television shows showed the work of Antonio Lanzavecchia and Rafael Baric. They are virologists and immunologists, and they were studying the SARS-CoV viruses in bats. So basically their idea was to study the viruses in these animals that are used or are very, let's say, people have promiscuous relationship with, because as you know, unfortunately, these animals are used in the Chinese culture, and they are also sold alive in the markets. And the contagion, remember, started from Wuhan, where there is a wet market where they sell these wild animals. So their research was going in the right direction. They were trying to study the virus in the wild animals and understand how they can jump the species and get to the humans. However, the research didn't receive the attention that it should have had. And actually, these scientists were criticized, and people started thinking that the virus was generated by them. So you can see how dangerous it is to spread this news and also feed the fear of people. A scientist at the end of this discussion concluded the virus was made by nature, and this up to the scientists now to show how nature could make this. So I would suggest always to, of course, read all the theories, read whatever people have to say, but of course, it's not possible to prove these theories right, so we shouldn't be alarmed by these theories, and we should just take them apart. There is something more important now to take care of. So I'm ready to discuss with you the scientific aspects of this disease and this virus. I hope we can have an open discussion, and any questions you have, I'm glad to reply. And also, I think after the presentation, it will be very nice to share papers so that you can really increase your knowledge about this situation and have your own ideas, because it is very important. In the next months and years, science will have to give us a lot of answers. And hopefully something like this will not happen for the next 120 years. This is the hope.
Thank you very much, Antonella.
Back to you, Mushraf.
Thank you very much. I appreciate the presentation. I appreciate the brief you gave for the students. Very informative. I like the final few slides where you talked about the possibility of inaccurate news, let's say, that might spread, and as scientists we should be able to scrutinize things to the T, as they say. Before I go forward, I'd like to welcome my friend. She's a colleague of mine. She studied with me in Manchester. Her name is Ahdab Saidi. She's a colleague of mine in the faculty, and she'll be part of our discussion. She'll jump in if she has any questions. I hope that's okay. إذا سمعتنا إهداب ما أدري صوتي واضح عندك إهداب أو لا؟ Well, she'll join us.
Hello?
Can you hear me, Ihdab?
Yeah, do you hear me?
Yes, we can hear you. Antonella, can you hear Ihdab? Is her voice?
Yes, I can.
Yes, hi, Antonella. First of all, thank you for this presentation. And we are sorry to hear the news from Italy. I do have a quick question about the viruses or the infection in Italy. It has been said that the analysis of the genetic sequence, when they isolated the DNA from the patients, it's revealed to be different from the one that's been isolated from China or from Middle East, and it tends to be very severe. It is associated with the appearance of severe cases in Italy. Do you want to talk about this and tell us more?
Yes, of course. I heard this news, but I think it hasn't been proven that the sequence is changed. You know, again, they started spreading, the virus is changing so quick in Italy. The mutations are on a daily basis. I haven't seen any papers proving these. And actually, I heard an eminent immunologist in Italy, Roberto Burioni, he said that the virus is not changing. It is the same that we find in China. So the mortality in Italy can be explained, the higher mortality can be explained probably with the fact that we are a very old population. So a lot of people are over 65 years old. And what we know is that these people are more susceptible to this disease, and this explains the high number of deaths.
Thank you, Ihdab. Can I come in on a comment, please, Antonella?
Sure.
Can you give us maybe an idea of what is COVID-19 in terms of immunology? From an immunologist, what is COVID-19?
So it is not something completely unknown. It is, let's say, a cousin of the virus that already caused SARS in 2002, if I'm right. So this virus shares similarities to a virus that we already know. Just it is different because it has jumped from a wild species. So basically it has adapted in a wild animal, and then possibly with contact with another species, has gained the property of mutating and being able to bind to human molecules. So we know the SARS-CoV-2 binds to ACE receptor in humans, and possibly this was the productive mutation that allowed the virus to jump from one species to another in the so-called zoonosis. What else we know? It is an RNA virus. So its genetic material is RNA. It is more difficult, we know, to create vaccines for viruses that have RNA as their genetic material.
In terms of the medium of we know it's the pangolin, and that's one piece of information that that Ihdab gave me a few weeks ago. In terms of the nature of the virus and how it induces the immune response, because most of our students and most of the listeners here might have, as I said, a basic capture of immunology. What is your natural immune response to a virus?
Okay, so in a physiological situation, we expect our innate immunity, so macrophages, granulocytes, provided they have a good HLA system working. They should be able to present antigens to cytotoxic lymphocytes and also CD4 lymphocytes. Dendritic cells also play a role. What is known that somehow IL-6, which is one of the central molecules produced by macrophages to attract lymphocytes, it is secreted in an exaggerated way. And it is also known that there is a protein called protein N in the virus SARS-CoV-2 that has a direct impact on the IL-6 promoter. So basically, when these macrophages, dendritic cells, encounter the virus, they mount an exaggerated response, which it's like it's causing confusion in the system, and also what we call a cytokine storm, where basically cytokines go a bit crazy, and rather than protecting you to mount the correct immune response, they generate lung damage in this case. And the cells that die are cleared out, but also they obstruct the alveoli. So subsequently, the power of these lungs to oxygenate the body is really failing dramatically.
And that's due to the damage, the secondary damage from fighting the virus. So you have a collapse of your lungs. Can you continue on that point as well? You were talking about the IL-6 promoter.
Should have gone more into detail. So let's say you have a code within each one of your cells where there are instructions for all proteins that have to be produced in order for the cell to live. So if we said IL-6 is a molecule which is produced by macrophages, dendritic cells, antigen-presenting cells, in order to mount a correct immune response, but we know that in physiological events, there is always a turn on and turn off phase. So how clever was the virus to basically invent that through this N protein, it can act indirectly onto the IL-6 promoter of the macrophage, so that their answer becomes uncontrolled. So basically, something which is usually good is exploited by the virus to create a confused situation where your immune system cannot protect you anymore. This is more or less how it works.
Which shows you how fascinating the viral intelligence is. Yes. Can we also elaborate a little bit more? I want to take you back to the innate immune response, Antonella. And this may be part of the basics where we want to brush up for the students before I take you more deeper on the cytokine level and what we know about the type 1 and type 3 family members. As I say, in most cases, your innate immune response in both hands, physical, chemical or biological, is more than efficient to fight your viral infection. How would you describe the ability of macrophages in facilitating an immune response against viruses solely on the basis of phagocytosis and then antigen presentation?
So, yeah, I think this is a war on two sides, basically, because epithelial cells, which are the main target of the virus, the epithelial lining of the respiratory tract, upper and lower, does express the ACE2 receptor. So in a first phase, you have infection of the mucosal surface. But then when the virus is able to replicate within these cells, and basically is present in the mucosal lining, the macrophages come over, but also because they express the ACE2 receptor, they also get infected. So I think they become a bit of like corrupt policemen. They came to help, but the epithelium is already infected. They are infected themselves, and they're not able to mount the correct immune response.
Do we know do we know that the functionality of the macrophages is reduced when infected? Have we seen any papers on that? I certainly
I haven't seen papers on that, but I would assume that they will find something like this, like redox capability of the macrophages. They will find something regarding this for sure, and I will not be surprised if they can find maybe that these macrophages get reprogrammed very quickly and very cleverly by the virus.
It will also be interesting to see how much of the infectivity rate is divided between the epidermis and the epithelium and immune cells as well, and exactly. is the ability of the virus to infect a macrophage or T cells for that matters, as I've seen in a paper a few days ago with Ahdab. Does that render the immune response paralyzed, where it can't respond in an efficient manner? Because our body is efficient in fighting viruses, and maybe that's one of the ways it fails. So, do we know of any other innate immune cells that are under the risk of this virus?
I would just like to add NK cells with CD8, as they are an important cells to mount an immune response against viral infection.
Okay. Can you, for the case of the students, Hadab, can you give us a brief about that as well?
Because it has been shown in SARS, for example, that the deficiency of NK cell is associated with more severe disease and the presence of SARS-specific antibodies.
So there is a paper called Breadth of Concomitant Immune Response in a patient, however, a non-severe case of COVID-19. We can send you the reference later. They looked onto the cells. I don't remember if they spoke about NK cells, but
they mentioned only one line about NK cells in that paper. So the line they showed was that NK cells in this case did not increase. Now whether this means that NK cells did not play a role or not.
The other SARS virus impair the NQ2Ds, and
so
it doesn't matter. I don't think the numbers matter, because sometimes in some situations they showed that the number is the same, but they do have an impaired function. So if you look at the number, it might. Yes,
that's an interesting point. And it's very hard to sort out and detect NK cells in general as well.
It has been shown in SARS in terms of the number, not of the functionality. So but it has been shown in other immune diseases, like autoimmune diseases, for example, like thyroid autoimmune diseases, that the regulatory T cell number, it might be decreased, but the important thing is that they do have an impaired function. That's why they can't really clear the effector immune cells in the thyroid.
Why do you think that we don't see it in this specific patient?
To be honest, I haven't came across a paper talking about NK cell in COVID-19 specifically, but they were trying to explore the role of NK cell.
I wanted to say, Hadab, the study about NK cells, I think now as samples will be available for research, I don't know exactly how does it work when a patient is hospitalized, how much can a doctor ask for a screening of cells, whatever. But also I think it's very important when they will do this kind of analysis, because we know that another SARS virus impaired the function of NK cells. It will be very important to cluster the population, basically population of the study also by age, because in an older age, of course, the functionality of these NK cells will be already impaired compared to a young individual. In fact, that paper that I cited, when I read it, I was a bit it gives hope that you can overcome the disease and so on, but you need to understand what happens in severe cases and immunocompromised people to understand what's going on there.
What's the diagnostic criteria? If we have a patient walking in, Antonella, in the lab, what tests do we do, and what are the accurate tests, and when does serology come into the picture, if you want to highlight?
So I was very curious about this because, as I said, I'm not a doctor. I wanted to be a doctor, actually. Now I regret that I'm not a doctor. So the main thing that worries the doctors when you come in, it's the blood oxygenation levels. So have you ever seen those things they put onto your finger in the hospital?
Yes.
So that tells you how well oxygenated your blood is.
So is that the step number one they take? Checking the oxygen levels?
Well, I don't know exactly, but in that video that I put in the slide, they explain all the clinical criteria. I don't know in which sequence, but one main thing that worries the clinicians is the low levels of blood oxygenation. And you know what's low level? So they consider low level even 92%. I was very surprised because 92 is a big number. So a good number is 98, 96. 96.
I wonder if that's different between age groups.
Probably there will be a difference because, for example, I did an experiment. I have one of those things at home. My blood oxygenation was 98, but already my mom's was 95. But it's still good. When you go lower than 92, that means your body is not receiving enough oxygen. So either I heard, I'm not entirely sure, that either they give intravenously something to amp up the oxygen levels, or they put you on the ventilation to help you. Then other clinical parameters, CRP, so C-reactive protein, gives you an idea of the inflammation that is going on in your body.
And it's one of the most profound markers for inflammation, C-reactive protein. So it's not strictly for viral infections.
Exactly, it's a broad one.
It's a broad one, exactly.
And I also heard that when there is a lot of damage in the lung, bacteria that reside in the lung can take over and multiply their numbers. And if they reach the bloodstream, you're basically getting into sepsis. Yeah. So there are different phases. There is the damage in the lung. Then if bacteria overcome, the body, they can create sepsis. And if the inflammatory response is all over your body, not localized anymore, that's very dangerous.
Do we know how sensitive we are in terms of PCRs being used?
Yeah, I think they're relying mostly on that. I heard about some tests circulating. They were looking into the IgM and IgG, but immediately they've been kind of put aside as not informative enough because, you can have immunoglobulins much later. So, of course, you need a PCR.
Please give us an idea of when the immunoglobulins are produced. So, because a lot of the questions that I get from the students are, why don't we look at the immunoglobulins straight away, purify them and send them back in? But the nature of the production of immunoglobulin is much later. It's a latent response rather than an immediate response. So can you give us a little bit of a brief on that?
Yeah, of course. We will find them much later after the immune response has been mounted. I think we are talking about 2-3 weeks. Something has been done in China to use the plasma of the healed patients to cure the patients in hospital. And I was hoping that in Italy we would have done the same, but there is a bit of secrecy about the procedures that they are following. And as Italians, we know that we are a bit poor. Probably if there is technology needed to clean the plasma, put it back in, I don't know if they're doing it systematically, but I think it would be a great idea.
Do we know of any other countries? You said China. Any other countries other than China that use it?
I don't know. I think France, close to us, is relying on chloroquine, which is an antimalaric. I haven't heard of any other country using the plasma. And I think we should know more about these things. Of course, we are not the doctors, but if there are options available which are used by other countries, why don't they consider them? And people want to know, are you trying this? Are you trying that?
As far as I know, that the immunoglobulin is produced late.
Yes.
Like, for example, IgM have been found in the serum of, I think, one patient. The study included one patient or more. I can't remember the number of patients.
Yeah.
But anyway, when they've checked, they found that the IgM has been peaked at day 9 after the infection. And by week 2, there is a switch, a class switch to IgG. So maybe this is not a very reliable to even the PCR, by the way. People with some symptoms tested negative for the COVID-19 PCR, but actually the CT scan and the other supportive things has shown like a sign of infection with SARS-CoV-2. To be honest, I think the incubation period of the virus, because it's like from 2 to 14 days, probably the viruses uses some mechanism of immune evasion until it has been detected by the...
Yeah, we brushed on that, me and Antonella, at the beginning of the talk.
Did you? Okay.
Yeah, at the beginning. So do you know, Hadab, what I know we've talked about this, me and you, before, but in terms of immune evasion of a virus, what would you think a virus can do?
To be honest, I'm not really sure, but I do believe that there is some immune evasion mechanism is involved due to the long incubation period of the virus. Afraid I came across only one review that we're talking about the possible evasion mechanism.
Can I ask you also, you said an important point before we open the panel for the questions from the students. You were saying how PCRs are not that sensitive and they still fail. And this is an open question for Antonella and Hadab as well. Is this due to the amount of viral load in a patient and it appears later or it appears earlier, or is it down to the lack of good PCR sensitivity kits?
I think it can be a combination of both. Well, the entry site for the virus should be the oropharynx, the nose. The swabs are done in the nose and in the throat. So either the virus hasn't replicated enough to be recognized with a PCR, or I think PCR tests should be quite standard, and I'm not entirely sure they're not sensitive enough.
I'd like to give a brief about the PCR. Maybe some of them have not practiced it yet. So with a PCR technique, we are looking for the genetic sequence that we have already identified of the virus. So early on in the epidemic, when it started, in the pandemic, when it started in a local market in Hubei, China, and Wuhan, China, scientists back then were very successful in identifying the genetic sequence, and they automatically, one of the earliest contributions, what they sent the genetic sequence to many labs around the world for people to generate identification kits or PCRs. With the PCR, we search in a given sample of a suspected patient for the viral sequence, for the genetic identifying sequence of this virus. And that's what PCRs do. We take a sample a swab sample from, as Antonella said, from the back of the throat. In that sample, we search for the genetic sequence that we have already identified for SARS-2. If I'm not mistaken, the PCR has been around for, is it 30, 40 years? So it's quite an established technique.
Yeah.
However, we do still struggle with the sensitivity, sometimes, especially with new viral infections.
I would also like to share a news that I came across, I think yesterday. I don't know if you have talked about this when I disappeared for 5 minutes, but there were 2 old guys or old people who survived the Spanish flu in 1920 2020 recovered from coronavirus, and they are very old. That was in Italy. So I don't know if this is true or not. And also,
Yeah, I
what do
think
you
it's
think?
true.
Yes, it is true. And they are
You,
very old. Like one
can
is
you
over
give us,
100 years
sorry,
old.
sorry, can you give us a brief on that? Because I think myself and most of the listeners might have not heard about this.
Yes, two old men, like one over 100 years old and the other one around 95 maybe, who survived the Spanish flu in 1920, the one of the previous epidemic, recovered from coronaviruses in Italy.
Mm-hmm.
So this is quite interesting because we know that the old people are at high risk of getting like the severe symptoms. So do you think this is due to their something related to the immune system, or just the luck? They were just lucky.
I don't know.
No.
I don't know.
Have they done anything? Like, have they do a lab investigation and any because I've just seen the news. I don't know, to be honest, any more details about these two patients. So I don't know if there is a case report about them.
So the Spanish flu hit Europe around the 1918, which was in the midst of the First World War. It killed up to 50, some people say just under the range of 100 million people, mostly young people. Do we know that you can be immunized against the Spanish flu and keep the immunization for 100 years?
Well, I don't think in 2018 they were doing this kind of tests. I think it is, of course, possible that they are immunized, but what are we going to do? Start testing all the old people?
Over 100.
Over 100, yeah, over 100. And I think these people are anyways so rare that possibly you wouldn't have a population big enough to draw any conclusion. But of course it would be worth testing. You know, when they show in the news, oh, he recovered, he's 100 years old. Yeah, they don't tell us the important things. Have they tested for antibodies against the Spanish flu, and are they protected because of these? You know, that they're not very interested in these, but they should be more.
Okay, thank you, Antonella. Let's give a chance to some of the students we have to ask some questions. A lot of interesting questions there, Antonella. I hope you're ready. And Hadab, please chip in. I've not seen the students this excited for a very long time. And when I say for a very long time, I mean, not even when I give them lectures, which is surprising, really. I think I'm very interesting. And none of you need to comment on that. Amira.
Okay, if people who survive, it will not catch it again, if there is any study about it or something.
So you're talking about immunization. If you are capable of clearing the primary infection, the first infection, can you clear the infection automatically through immunization the second time? So do you have memory cells?
So yes, the hope is the patient can fight the infection through its own immune system. But unfortunately, there are also reports, again, I invite you to confute them and read from more sources. Some people also tested positive a second time. So there's something that it's not completely clear.
I think this is because of the high recombination rate of this virus, because the recombination rate is very high in this virus. So if you got the same virus, okay, but it's highly mutated. So that's why it's like the flu virus.
Is this the case in the flu virus, Ahdab? If you get tested once, if you get tested again in a matter of weeks or months, you will not test positive for the same. Is it that highly mutating?
Yes, I think so. The high mutation rate. That's why even if we do have a vaccine against the flu virus,
you can get it.
Each year, yes, you can still get it. And each year we do have like a different vaccine against flu virus. The 2017 and 2000. Each time they found like a new epitopes or something. Yes. So the high mutation and recombination rate of the virus is one of the problems that you might get it again.
A lot of people are asking, is there a possibility of a genetic predisposition in Italians? that led to this outbreak on this scale in Italy, unbelievable scale in Italy. Do you think, thinking outside of the box, we know there are environmental factors that led to it. You were talking about the lack of response, the lagging from the authorities. Is there a genetic predisposition that has been entertained or talked about in the Italian media or in the Italian scientific community?
No one talked about this yet, not that I heard of. But I've been asking these questions to myself as well. And one thing that I said with a smile on my face, not believing 100% what I said to myself. As I said, North is the region where there is most productivity. People have a fast life compared to the south of Italy. And I was thinking, is it not that maybe a lot of people are, they take drugs for hypertension, so this problem with the ACE2 receptor, but I don't know. And I think maybe most scientists would say, no, there is no genetic predisposition.
I would like to give more students a chance to ask a question. There's Mohammed Suleimani. Mohammed, can you hear me?
Yes, I can hear you.
What's your question, Mohammed, to Antonella?
When the first news about the coronavirus outbreak came through, it was reported that it affected almost everybody. But with the disease progression, news about it only affecting older people, and children are not that much affected by the virus. There is a reason for that.
Thank you, Mohammed. That's a very good question.
So in Italy, we had no cases of children. The youngest person who died was 26 years old. So I give as explanation what I said before. The only theory I can think of is the vaccinations that the children are exposed to very early in life. And if you compare a child to an older person that maybe is in his 70s and hasn't had a booster for years, probably what I can think of, the innate immune system is trained by these vaccinations to mount a more solid immune response.
There's no really clear understanding why children are less infected. They were talking about, brushing on what you were saying, Antonella, that children have been exposed to so much as they're building their immune archive. So they are, let's say they've got the bullet in the gun already. Therefore, they are able to deal with new unfolding infections better than adults. But yes, it's a big question mark, which is definitely worth many PhDs to go on. Adab?
Sorry, what was the question?
So they were asking about why do we think that children are not getting the infection?
This is interesting to be explored, actually. What is the difference between why the elderly people or even the young people? It has been shown like one children with an immunocompromised, immune deficiency has got the infection, as far as I know.
What type of immune deficiency? Overall?
Overall, it has been shown that the child has always gotten infected, and it has a weak immune system, and it has got the virus. It had tested positive. I think that was in Kuwait.
Chef, I wanted to add, sorry, that it has been said somewhere, I don't have the reference though, that also children do express lower levels of ACE2. So this could be also another reason.
That would be logical, yeah.
That would make sense 100%. I think, and that falls on the heel of maybe that can answer the question we had about ibuprofen and hypertension. So that can definitely fall on in that bracket. I want to allow some more students to ask. People are getting rowdy, I think, on the chat. So let's give a chance to Rahaf Al-Sufi, and then Asil can follow her. Rahaf, can you hear me?
Yeah, I can. Hi.
Hi.
I was talking in terms of the virus. When it encounters so many people and different immune responses, could that possibly create a resistance for the virus? So we can have like a pandemic resistance of the virus, that nothing could defeat it anymore. That's what I kind of meant.
So what you're suggesting is rather than us forming an immunity, the virus mutates enough that it protects itself. And we
all sorts of medication, treatments, and immune system in general.
If those mutations occur, to make it resistant to whatever treatment we are going to put in place, I think we will be stronger than this.
Asil, what's your question?
There are many people who have recovered from the disease. Well, there will be a memory cell. Would this help us to know it's all properties of a virus?
So again, the question about memory cells, Antonella.
Yeah. Can you repeat the last bit, because I couldn't hear it?
Okay, we have, of course, a memory cell for the virus, for the people who recover.
Okay.
Can we know all the properties of the virus from it?
That's a very interesting question.
Yeah, I think it's a very good point. However, already from the genetic material, hopefully with bioinformatic tools and proteomics, we should be able to learn enough about the virus. The antibodies, I think they would basically be helpful for that plasma operation. So using the antibodies of a healed person to treat another one.
Thank you. Wid, what's your question, Wid?
Yeah, I was asking if we can do a receptor blocker for the virus, so the virus cannot bind with the lung cells.
That's a very good question. I used to work on a monoclonal antibody program, and I was so impressed. How long does it take to generate a monoclonal antibody? You have to have your hybridoma in a cell line that you choose. Then there are many aspects of the development which really represent a challenge. I don't think in a critical situation like this is something that will be done quickly. We will get there, but it is not something that we expect to have in the beds of the hospitals in 2 weeks' time. Okay, also, I thought about an important thing. So if the virus is entering through the respiratory tract, and you basically have the virus replicating mainly in the lung, I also read a paper where they claim coronavirus cannot be detected in the blood. So this virus is replicating selectively there in that location. What do? Monoclonal antibody, usually you do intravenously, intraperitoneally, whatever. Can you do an inhaled monoclonal antibody? That would be a very critical challenge for development.
Can you give our students a little brief before I move to the second question? The difference between a monoclonal antibody and the more generate known polyclonal antibody. In brief, Antonella, so I can move to the second question.
Yeah, perfect. So usually monoclonal antibodies are referred to as more specific antibodies because they are selected from a single clone of hybridoma cells or B cells that produce these antibodies. Whereas polyclonal antibodies come from a mixture of clones that produce antibodies, and they will be possibly specific for different portions of the antigen, but we refer to them always as less specific than a monoclonal one, which can target selectively only one portion of the antigen in a very specific way.
Thank you, Antonella. On the heel of your question, there are protease inhibitors that are being in trial now to try to block protease components on the surface of epithelial cells, which are very important for viral shedding and viral infiltration of a cell. So not only do we need to target the ACE molecules on the surface or the spikes, but we need also to address other components found on target cells to prevent viral infection. Faris, can you hear me?
Yes, I can.
Okay, Faris.
I had actually a number of questions, but all they were answered.
Okay.
So there is one left question from one from my friend in the group. If the coronavirus can transmitted by blood transfusion, can the immune system response more affected?
Say the question again, Faris, please.
If coronavirus can transmitted by blood transfusion, can the immune response to this coronavirus?
Interesting.
Thank you, Faris.
Yeah, that's it.
So you mean if the virus gets transmitted through blood, can you have a better immune response?
Yes.
That's a very good point. But I don't think there will be any volunteer that wants to get the coronavirus in the blood.
I can see some hands raising. Maybe they mean they want to volunteer. I'm not sure they want to volunteer for this, but they have questions. Thank you, Faris. A very good question. Because Mohammed, you asked the question, let's give a chance to Rima. Rima Abdullah, what's your question?
Hi, I'm Rima.
Hi, Rima. What's your question?
My question is: malaria patient immunized against COVID-19 due to use the same anti-malaria drug for corona patients?
Yes.
Or there is no relation?
Very, very good question. Very good questions. What do you think?
That's a very good question.
Very good question, Rima.
Yeah, and I hope, well, unfortunately, the malaric area, I think it's Africa. I don't know if, well, the virus got there, but I don't know if they will have the chance to monitor patients that are taking anti-malaric or they have actively malaria at the moment. As far as I know, the chloroquine treatment helps to reduce viral replication. There is a study where they show in a patient, a COVID-19 patient that is taking hydroxychloroquine, the viral load reduces. So it has something to do with viral replication, interfere with the machinery for replication. Unfortunately, in Italy, they're not using as actively as in France, and I don't know the reason for this. So, but it would be really interesting to monitor patients with malaria and also see if the propedeutic treatment, prophylactic treatment helps.
Yeah, so we don't have any data for patients that have malaria and have been infected with corona.
I don't know. We need to
unfortunate
search. Maybe there are.
Well, yes, something will come up, but the unfortunate circumstance of having enough to be scientifically and statistically relevant. It's been my personal pleasure. I've known you for years. You're one of the smartest people I know in terms of understanding immunology, and it's been a pleasure to have you introduce you to these young and hopefully upcoming future scientists who want to know more about this. Do you have any words of encouragement to these young students?
I think the future is really hard but bright ahead. As a scientist myself, sometimes I questioned, oh, am I needed in this world? I think this is the moment. There is something we need to understand, and importantly, not to allow anything like this to temporarily block our lives. I mean, we had wars. This is even... those are even worse events, but many people died, and it's really sad. So scientists will have to help understanding what happened.
Well, again, I'd like to say thank you. Ahdab, if you're still around, if you wanted to add a few words to sum up this very interesting meeting we've had with Antonella.
Yes, I just would like to thank Antonella for her time and for the discussion. It was very nice and interesting, stimulating discussion. I don't have anything to add at the moment. We're still waiting the research coming up to understand more the relationship between the immune responses and the disease pathogenesis. But also I'm interested to see how the viruses adapt from animal to human as well, because, for example, in bats, it's not very I know that it adapts very well in bat with no severe diseases or even a sign of infection. So I think people are studying this as well, if I'm not wrong. So this is the only thing I would like to add.
Yes, well, only the future will show us. Everybody, thank you. Everybody, I'd like to thank every and each one of you for participating. I hope this keeps you eager. We've started this to keep the ball rolling. I know we spend a lot of time now in quarantine, but hopefully this will be an educated 2 hours that we've spent together. Antonella, ci vediamo. Ricardo.
Thank you very much. It was an honor.
All right, take care. Thank you very much.
Thank you. Bye-bye.
Thank you, and bye-bye, everybody. Thank you. Bye-bye.